trainer one's data. What we are going to show now is a tool called OMERO.parade, which is a data mining tool, and we will show how to go through the images which are stored in OMERO based on the qualities of the images, such as came from the results tables coming ultimately from our analysis in Fiji. Okay, so if we select the project called IDR 0021, then I already promised that the AFiji dataset, which we were working on before the break, is just like kind of best off from the full study, and the study is the IDR 0021 published images which are super resolution confocal super resolution microscopy of the centrioles, so subcellular structures. Okay, and we have run using the Fiji script, which we were plentifully demonstrating, the segmentation on all 414 of them. Okay, this resulted in an OMERO table, and the OMERO table is here as an attachment. Furthermore, we produced a CSV, a comma-separated value, things which you can open in OMERO Excel, as in Excel, of course, in Microsoft Excel, and just to show that you can do both. Okay, just it's a redundant thing, but it will show the power of the system. And now when I click on the I on the attachments tab on the summary from Fiji, I can study the OMERO table, which was produced in Fiji. Now let me you can do some querying of the table like so. So it just shows that where the count is greater than 400. Okay, so this is some parameter coming from Fiji. Okay, and I can filter inside the OMERO table. That's great, but OMERO.parade can do much better than that. Let me just allow a small aside. One other thing, one other advantage which the OMERO table gives you is that you can, in the web client, when you navigate to a single image inside that portfolio, you can click on the tables tab and you can see that the values which are pertaining for this image from that OMERO table are displayed under tables tab. So basically, the OMERO table has quite a few columns, and each column here is displayed as a row, and the values from the row which pertains to the image number 5 from the CDK for RAP dataset are displayed in this tab. Yeah, that's the image I'm selecting. So in this way, you can also add metadata to your images. Even if you would not use OMERO.parade for data mining, this can be quite handy. And we also ship a script which will do that for you using a UI. You don't have to bother with Fiji. Let's say you have your data as a CSV. And if you would like to turn your CSV into a OMERO table, you can do it using this script, which is under import scripts, and it's called Populate Metadata. And if you click on that, you can see, you can select a, let's say, data set, and on, if I would have been selected, if a full data set here would be selected, then it would pre-populate. Let's do it. I select, let's say, a data set called SVS, and I can populate metadata. You can see that the ID of this dataset is pre-populated in that script, and now the attachment is not there. Okay, so I don't have any CSV attachment attached, but it doesn't matter. I can browse and I can say upload the CSV, let's say this one, and it will do it for me, and then I will run the script which will create some tables, and then it will display them as values here. So we've done, you can do that as well. Okay, if the CSV has certain qualities which you can find described in the OMERO guide if you search in the OMERO guides for populate metadata. Populate metadata. Okay, so you go to OMERO guide, search for that, and you will find how to create an OMERO table in a more easy way than when using Fiji. Good. But back to what we wanted to show, and this is OMERO.parade. So we're having the table, or indeed having just a simple CSV. So OMERO.parade now swallows both. We can simply click on the super container, which is the project in this case, and select OMERO.parade in the central pane from the drop-down menu just to the right-hand side above the central pane. And if we do that, we can see if I close the dataset on the left-hand side, no datasets are open, and thus the OMERO.parade does not show anything. It just works on the open dataset. I can go open all, and it will open all the images in all 10 datasets, all 414 of them. I can add now filter. For example, I can filter them by regions of interest count. So how many regions of interest did Fiji draw on each of those images? I can fathom it out like that. I can say the regions of interest number on that image must be bigger than 20, but it must be smaller than 50. And here I go. I have the images here filtered out, which have image regions of interest numbers such as 35 or 48, which is definitely between 20 and 50. Okay, I can remove this filter, and I can go ahead and say I want to show a table view of those thumbnails in the center pane, and I can add table data into the table view. So let's say you can see here that I can add data from the CSV, but I can add data from the OMERO table as well. As indicated, the OMERO table has one row per each image, and so does the CSV. I can add the table total area, which is the area of the biggest region of interest in that image, as a column like so. And I can also add a table bounding box, which is the area of the bounding box, which encompasses the biggest region in the image. And then I will add simply table image, which is the ID of the image. Having these three columns like so, I can now check the checkbox, which will simply switch on the color coding on the rows, and I can do it for all three and sort the data according to the value in this column. And I can do it per data set, or I can do it for all. These images have simply no values in the CSV, and I can now.
Peter, sorry, can you show again how to add more filter? Christina missed that.
Yes, so I am adding filter. Okay, so here. So for example, I can filter for.
But you're in this as the middle. You're in the middle tab already. Is it? Or you just? Yeah, continue. Sorry.
So filter addition is here, okay? So I can do that still. I can still add here a filter, and I can go between 20. You see, so I'm filtering also, doesn't matter that I'm already inside the tab, and I can add another filter for smaller than 50 regions of interest, okay? And now I can, oh, okay, got it. Okay, yeah, exactly. You have to always drop down the menu again and again. And you can go by rating, okay? Rating 2, and this will filter them all out because none of them has any rating, yeah, etc., etc. That's fine. I know that this might be slightly, the controls here are a little bit over the top. OMERO.parade is still kind of in its infancy, and this is not completely, how to say, matured in the usability sense. That I'm using another menu to add new columns into this view. Okay, so this is the lower menu, which is saying add table data. But I have added already my 3 columns, and you can see the sorting capabilities of here, and I can already see some patterns here on this heat map coded rows for those 3 parameters. Yeah, so if I, let's say, sort according to total area, I can see that these ones have the biggest total area, etc., etc. And very interestingly, I have here a graph icon on the top right, and if I click on that small graph icon, I can go and see a graph. Each point in this graph represents an image. And if I swap the axis here, let's say like this, like so, I have table total area on the y-axis, and on the x-axis I have table bounding box. And because I know what the parameter mean, then I am quite unnerved by the 2 outliers here, which have such big bounding box compared to their area. So I would like to know if my automatic analysis in Fiji really produce sensible regions of interest for those 2 images, or if indeed they are invalid. For that, I would open those 2 images in OMERO iViewer, like so. Here is an open with menu, or yeah, so I can click on that. And this is another power of OMERO iViewer. It can open any images, any collection of images which you select in the tree in the left-hand side, or indeed using OMERO.parade as I just did. So it doesn't have to be from the same data set. And I can inspect the regions of interest here. And because I was telling you that in those values, it's always the biggest region of interest on the image which is gonna contribute to the to the statistics and evaluation, I can see immediately that this biggest region in this image is invalid. It does not encompass the 2 centrioles in the image, like so. This would be 1 centriole, this would be another centriole. Instead, it's a huge region of interest of the shape of a big sausage, and has 2 unwished for whiskers here, and it encompasses both of the centrioles. So this didn't go well for this image for Fiji, and I can discard this ROI from my evaluation. So you can see the power of OMERO.parade as a quality assurance tool. Good. Now, let me just show you another tool on this node, which we have in our portfolio, and this is called OMERO Figure. The OMERO Figure is probably our most popular tool in OMERO. It allows you to create publication figures for presenting in a scientific journal, but even more modestly, if you just want to show data quickly to your boss or on a lab meeting, then indeed you can do figure creation in this very quick manner, which we will just demonstrate as we go. Before we do that, though, because we have a little bit time of spare, let me show you one more power of the OMERO.parade, which will bind nicely to our previous workflow with Cell Profiler. So you remember that we were working with Cell Profiler on this plate, and we were analyzing basically 5 wells out of this 96 well plate. Which is great, but we would like to analyze it all. Why didn't we do that? Well, because of time reasons, obviously, but we did it already for you previously. So we have the OMERO table here for that plate. This was done by Cell Profiler, again, viewing the OMERO table on the plate called IDR00, no, pardon me, plate called plate 01 underscores 1 underscore 13 underscore previously analyzed, as I'm highlighting here on the bottom left. Okay, now I can drag it. Very good. So you can see the name plate 1 underscore 1 underscore 013 previously analyzed in the data of trainer 1. That's what I'm playing with now. So I zoom it out so that I can see all the wells. In the plate, I already showed you the attached table, which comes from previous analysis. And I can now switch on OMERO.parade again and say, I want to add filter from the table obviously being this one, this thing. So I can filter in the table. Now in the table, in the OMERO table, there can be strings, meaning non-numerical values, like text values, but there can also be numerical values such as cell count, which is probably the most human understandable parameter. It's simply the number of the cells which were detected by cell profiler in this particular well. Okay, so if I now start dragging the slider, you can see that the wells which have now a lower cell count than the 35, which is highlighted here, okay, are vanishing, whereas the ones which fulfill the criteria are still being shown. Okay, I can of course, again, as you asked, I can of course add the cell count again, as we did with the Royce in the previous example, and it will be bigger than 35, but smaller than 40, then 45, 46, smaller than 46, so bigger than 33 and smaller than 46. Okay, so that's where I am in between. And if I, let's say, remove this filter, then I can go and show only the ones which have exactly 33 cells in them. And you can see here in the tables, if I select such well, which I filtered for, if you click on the tables, by selecting this well, you can see that there is on the bottom a cell count, and it says indeed 33. Unsurprisingly, because both these this content of this harmonica as well as the OMERO.parade are picking data from the same place, and the place is the attached OMERO table. Very good. So much for the OMERO.parade.